Alright, hello everyone. So we're going to continue on in part 3 here, and we're going to build off of where we left off. So we left off talking about how to make carbohydrates. An additional major component of the cell are lipids. So we need to be able to make membranes if we're going to make a new cell. And so let's take a look at how lipids are made. So, if we think about lipids, essentially it requires synthesizing fatty acids, and we'll see that that is essentially beta oxidation in reverse, and it requires making glycerol phosphate. And so glycerol phosphate is something we can make readily from gluconeogenesis or right off of glycolysis, and it's actually one of the most expensive macromolecules to make. And so if we just kind of conceptually think about how much energy was generated when we talked about beta oxidation and oxidizing a fatty acid, you can't get around thermodynamics, and you have to pay the same energetic cost to make things just as you gain energy from breaking them. So let's take a look at how this gets done. So fatty acid biosynthesis is actually a really elegant process, and it's elegant in the same way that beta oxidation is elegant. And unlike beta oxidation, which uses acetyl-CoA as a carrier, essentially we are going to use the acyl carrier pathway, and we'll see what happens with this. So you use acetyl-CoA to build lipids, and so we saw in beta oxidation that we produce a ton of acetyl-CoA from fatty acids, but we can use acetyl-CoA to build lipids. So what about malonyl-CoA? So when we look up here, and I'll get to what these ACP things are in just a second, but you need to condense acetyl-CoA and malonyl-CoA to initiate this whole process. And so malonyl-CoA is essentially generated from acetyl-CoA by addition of CO2. And so you might say, well, isn't this autotrophy? The short answer is no. This is what's known as an anaplerotic reaction, and actually the CO2 that gets added on ultimately gets dumped off during this condensation reaction. So just like when we needed to basically turn pyruvate into a 4-carbon substrate and then back into a 3-carbon substrate, the same sort of carboxylation and decarboxylation is used to condense acetyl ACP and malonyl ACP, and I'll get to what that means in just a second. So, unlike the beta oxidation pathway, which uses CoA as a carrier handle essentially to bring these fatty acids through oxidation, instead what the acyl carrier pathway uses is uncreatively acyl carrier protein. And so it is like CoA in many ways in that it's a large molecular handle that gets added on to a very small substrate, in the case here acetate more or less, and allows enzymes to interact very readily with this compound. And additionally, ACP versus coenzyme A allows for acetyl ACP to be preferentially used for this acyl carrier pathway for building fatty acids and not oxidizing them. So again, this is kind of like what we're going to see with NADPH in just a second when I'm talking about this, and when we talked about D-amino acids in the cell wall, and we talked about gluconeogenic reactions versus the oxidative reactions of glycolysis, that essentially the cell is looking to partition processes in very fundamental ways, so that when you're building things, you're building things, and when you're oxidizing things, you're oxidizing things. And one of the ways this gets done is through the use of different molecular handles, like acyl carrier protein instead of coenzyme A. And so again, this allows for partitioning of catabolic, oxidation, type reactions and anabolic or biosynthetic reactions. Okay, so what happens in this fatty acid biosynthetic pathway is that acetyl ACP, so essentially acetyl-CoA has an exchange reaction that adds on acyl carrier protein in its place, and same thing with malonyl-CoA ends up with ACP on it, and condensation of acetyl ACP and malonyl ACP produces acetoacetyl-CoA, and you might remember this, if this were CoA, looks like an intermediate from beta oxidation, right? So now what happens is two NADPH, essentially two reaction steps that use an NADPH each, reduce this ketone down to a just alkane chain here. And what can happen, so now we have a 4-carbon fatty acid. So now we can repeat this same process with butyryl ACP and add on essentially another malonyl ACP, and you just keep doing this over and over and over, and every time you're adding on 2 carbons. So we can end up doing a little bit of math that we're going to do later about how expensive is this process for the cell in terms of NADPH that needs to get spent. So if we want to think about how much NADPH is required for a 16-carbon fatty acid like we just saw, that ultimately 14 NADPH are required and 14 ATP. So 1 ATP per malonyl-CoA produced from acetyl-CoA. So I'm not telling you I'm not showing the biochemistry of that. That is, it's food for a different course. But know that every time that you need to make a malonyl-CoA, you're going to need to spend an ATP. So minus the 1 acetyl-CoA that you brought together with malonyl-CoA in the first reaction, you can kind of think of how many carbons is the fatty acid minus 2, and that essentially tells you how many NADPH and how many ATP are required for producing that one fatty acid. So remember that phosphoglycerol or glycerol phosphate lipids, or phospholipids, they have a glycerol phosphate backbone, remember? So attached to glycerol is a couple of fatty acids, and that there's a phosphate group on glycerol. So where did those come from? Well, it turns out that the aldolase reaction in glycolysis that we talked about produces 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. Now, this is actually a really important partitioning reaction for the cell, because remember, 3-phosphoglyceraldehyde continues down through glycolysis, and dihydroxyacetone phosphate actually is a branch point to produce glycerol 3-phosphate that can go on and be used in lipid biosynthesis. So again, this is one of these events in glycolysis that's allowing essentially metabolic decisions to be made. If the cell needs to make lipids, it will take dihydroxyacetone phosphate and make lipids at the expense of oxidizing this dihydroxyacetone phosphate through glycolysis to generate more energy. So essentially you're spending the energy indirectly now to go make cellular constituents. So if we're going to put all these pieces together, you need glycolysis to prove it. If we're talking about making lipids from growing on glucose, we're going to end up with glyceraldehyde 3-phosphate and dihydroxyacetone phosphate during glycolysis. Dihydroxyacetone phosphate provides the glycerol backbone. Now we're going to oxidize essentially glucose down to acetyl-CoA, which can go into the Krebs cycle or the citric acid cycle, or these acetyl-CoAs can be put into the acyl carrier pathway to produce fatty acids. These two, the glycerol and the fatty acids, are condensed to produce lipids. So let's do a little bit of accounting. So if we are going to think about palmitic acid again, that C16 fatty acid costs the cell 4 NADPH and 14 ATP. So essentially the number of carbons minus the 2 carbons you got for free from acetyl-CoA. Okay? So each dihydroxyacetone phosphate now requires 1 NADPH to produce that glycerol phosphate that's going to be essentially the phosphate head of the lipid, right? So then each lipid has 2 fatty acids and 1 glycerol phosphate, so it requires 28 NADPH, 28 ATP, and 1 NADH to make a lipid. Okay, so that takes us to what is required to make lipids. So the pathways you need the acyl carrier pathway to make fatty acids. You need the citric acid cycle to either produce acetyl-CoA indirectly, or glycolysis that you can produce acetyl-CoA, or you need gluconeogenesis to produce glycerol, or you can get glycerol through glycolysis. So what I'm showing here mostly are the reverse reactions like gluconeogenesis to produce things. All right. So of the 4 major constituents of a cell, now we want to look at proteins, or essentially amino acids, right? So how are we going to get amino acids from different carbon compounds? So, as we've already seen, amino acids are produced from existing 3, 4, 5, and 7 carbon skeletons already being made by the cell. The only important step is adding the amine group. Okay, so just like we took them off earlier to eat amino acids, now we need to put them on to make them. So, the sources again of these carbon skeletons for amino acids. So the alanine, we can basically break these down into structural families, and we can easily see where these come from. So alanine and serine, essentially these are products of glycolysis, and to produce aromatic amino acids, you need phosphoenol pyruvate and erythrose 4-phosphate from the pentose phosphate pathway, so essentially a 3-carbon compound of 4-carbon sugar phosphate. You can bring these together to make aromatic amino acids. So then if we want to make the glutamate family amino acids, so proline, glutamine, arginine, and glutamate, these are derived from alpha-ketoglutarate, which remember is an intermediate in the citric acid cycle. It's a 5-carbon intermediate. Or the aspartate family, which includes aspartate, asparagine, lysine, methionine, threonine, and isoleucine. But these are derived from 4-carbon sugars, and this is the result of using oxaloacetate. Okay, so I think it's fair for me to ask you, if where are these alanine family amino acids coming from? They're coming from pyruvate. Serine family amino acids, they're coming from phosphoglycerate, and so on. Okay? All right. So addition of amines to carbon skeletons, this can happen a couple of different ways. So we can do direct amination via dehydrogenase. So this expends NADH or NADPH, and this is ultimately very specific for amino acids. And so we saw this for removing amine groups earlier. Or you can do what's known as reductive transamination, and we've already seen this, except it's going to operate in the opposite direction. So this requires NADH and ATP, and it works for all amino acids. So ultimately, both of these processes happen, but ultimately this reductive transamination process works for all amino acids, and is conserved not only in bacteria and archaea, but it's conserved in all living things. And so this is one of the ways that ammonia, nitrogen, is assimilated into cells. And what I'm showing here is the first step of reductive transamination, known as GOGAT. And so it's essentially an old acronym for essentially glutamate synthetase and glutamate synthase reactions that are happening. So glutamate is actually an alpha-ketoglutarate. These are incredibly important compounds in cells in many ways because we need to be performing this reaction to essentially get amine groups into this important amino acid pool. So we have free ammonia. ATP is spent to put ammonia onto glutamate to make glutamine. Glutamine is then a donor, an amine donor to alpha-ketoglutarate that will produce a glutamate. Okay, and this requires the expense of NADPH. And so this is how reduced nitrogen is incorporated into biomass by nearly all prokaryotes and eukaryotes. So when we bring in nitrogen, if we're just bringing in free nitrogen from the environment in our diet, this is how this is happening. That it's being put onto amino acids and transferred to other amino acids. So now this is where things become really very, very elegant. And so once you have aminated alpha-ketoglutarate via the GOGAT mechanism that I talked about, now we have glutamate, and there are a number of transaminase enzymes. So here I'm showing how you would make valine, and it could be tyrosine, it could be any amino acid that there is a transaminase that will use glutamate as a substrate and use glutamate as an amine donor to add amines to any carbon skeleton like alpha-oxoisovalerate, which is the precursor to making valine. Okay? Alright, so the major observations of amino acid biosynthesis are that precursors come from intermediates of glycolysis, the pentose phosphate pathway, and the citric acid cycle. Precursors are alpha-ketocarboxylic acids, so think alpha-ketoglutarate. So these are these carbon skeletons that are used to make amino acids. And most amino acids are produced by reductive transamination. So this is an important process that has two steps. One, the addition of amines to glutamate to make glutamine, and then transfer of amines from glutamine to an amino acid precursor. Okay? So production of glutamate is essential to making all 20 amino acids and costs the cell about 1 ATP and 1 NADPH to produce an amino acid from associated precursors. Okay? So it's actually fairly cheap to make amino acids, once you're accounting for kind of all of the carbon building that needs to happen compared to what we just saw for lipids, which are very, very expensive. All right, so proteins, what you need to make them, you need the citric acid, or amino acids, you need the citric acid cycle, you need the pentose phosphate pathway, you need glycolysis or gluconeogenesis to produce 3 carbon intermediates, and you need reductive transamination to add amines. All right, so where we're going to wrap things up is talking about nucleotides. So nucleotides, they're composed of 2 things. One, a sugar phosphate backbone that's made of ribose 5-phosphate. This is again produced by the pentose phosphate pathway. And nitrogenous bases. And these are produced using amino acids and single carbons carried on folic acid. And this will make sense in just a minute. So there are 2 families of bases. The first are pyrimidines. And so pyrimidines are derived from ammonia that is incorporated into the cell, oftentimes by reductive transamination of other compounds. Aspartate is essentially a large piece of this nitrogenous base backbone, and CO2 that is essentially fixed into this molecule. Again, this is not autotrophy. This CO2 was deposited onto onto this nitrogenous base via carriage on folic acid. And so this is one of the reasons that folic acid is actually, and is an essential nutrient in our diet, and is important for essentially synthesizing nitrogenous bases to make DNA and RNA. Okay. And then we have purines, which are larger, and that these are adenosine and guanine bases. And so where do all of these pieces come from? So again, we have these formal groups, and they're coming from folic acid, essentially. We have an amino group from aspartate, again, CO2 incorporated, and glycine. So ultimately, we have a bunch of amino acid constituents. We have things from folic acid incorporating things. And so I'm not going to get into all of the biochemistry of making those rings. I just want you to be aware that there are places that these things come from. So if we're going to make nucleotides, we need the pentose phosphate pathway for making ribose, and more or less what I want you to take away from making nucleotides is you need to be able to make amino acids like aspartate to make these nitrogenous bases. Okay, so the nitrogens that are in those bases, those are coming from amino acids. Okay? All right. So one of the things, if I were to summarize what we've seen so far, is that NADPH is the currency of biosynthesis. So when we're oxidizing things and we wanted to use respiratory chains and had NADH dehydrogenase, for example, complex 1 in our electron transport chain, all of those enzymes are using NADH, okay? So all of the enzymes that are biosynthetic that we've looked at, by and large, use NADPH as a substrate to provide reducing power to reduce oxidized carbon compounds to make more complex carbon compounds. And so again, this is one of the ways that cells make certain catabolic and anabolic reactions that share the same chemistry, use different enzymes. Okay? So this is really, really critically important that, again, the cell is partitioning catabolic and anabolic processes so that they can operate simultaneously, and it's essentially the great balancing act of molecular life, that you are oxidizing things at the same time that you're reducing them to essentially exchange certain types of carbon to make different types of carbon compounds so that you can eat one thing and produce everything you need to produce new cells. And that is what bacteria and prokaryotes in general are just amazing at doing. And so when we want to ask, where is NADPH produced? We've really only seen a couple of places that this gets produced. And I've kind of tried to highlight this along the way because this statement, NADPH is the currency of biosynthesis, is a little bit confusing when we've only seen 2 places so far that NADPH gets made directly. And that is through the pentose phosphate pathway and the Entner-Duderoff pathway, which we would only find in prokaryotes. So what is interesting is that ATP is expended in reactions that are biosynthetic, but nowhere near as much as NADPH. I apologize for the typo. So what does this tell us? So this tells us we oxidize things, we're making NADH, the cell's using electron transport to generate a whole bunch of electron transport and oxidative phosphorylation to generate a lot of ATP. And so, but ATP is not NADPH, right? So let's take a look at how this conundrum is dealt with. And how it's dealt with, actually, is a really marvelous enzyme called transhydrogenase. And so what happens is when we've been talking a lot about theoretical yields from glucose. So glucose being oxidized all the way down to CO2, all of those NADH that are produced transferring their electrons into an electron transport chain to generate proton motive force. If all of those protons went through ATP synthase, we can imagine we'll get, 32 ATP per glucose, right? Depending on coupling number, depending on a number of other factors. So if we want to actually think more strictly about cellular accounting, is that there are other things that are needed. So you need a lot of NADPH, especially for making lipids, right? We needed, for a 16 carbon fatty acid, you needed 14 NADPH. If you want to make that whole lipid, you need 14 NADPHs to do that, or 28 NADPHs to do that. So now what I'm going to do instead is do something tricky if I'm a prokaryote, for example, and I'm going to use a membrane-bound complex called transhydrogenase that is going to use the translocation of protons to essentially exchange electrons between NADH and NADP to produce NADPH. And so this transfer of electrons, because the electropotential between NADPH and NADH is just subtly different, that if I want to make this exchange, I have to spend protons from my proton motive force to produce this NADPH from NADH. Now this is a worthwhile reaction to do because you need all this NADPH to produce fatty acids, to produce amino acids, to produce nitrogenous bases. You name it, biosynthetic reactions, they need this. So what prokaryotes are doing, and we also do this as well, is that they're expending some of the proton motive force that's sacrificing ATP yield to produce NADPH that's required to perform biosynthetic reactions. So, that said, let's summarize. So NADPH is the currency of biosynthesis. It allows for simultaneous regulation of catabolic and anabolic processes. This makes certain catabolic and anabolic reactions that share the same chemistry use different enzymes. So there are enzymes that will use NADH, and there are enzymes that are going to use NADPH, and that they exclusively use one or the other. So NADH is only produced in a couple of places naturally, and we've seen this done in a couple of pathways. So I hope this actually makes the Entner-Duderoff pathway make a little more sense, right? Like, it didn't make a lot of sense that you were sacrificing ATP yield to produce NADPH. But what prokaryotes are doing that perform this glycolytic pathway is essentially speeding up the process. And ultimately also, if they're fermenting glucose and using the Entner-Duderoff pathway, they don't need to rely entirely on generating proton motive force to use transhydrogenase. So you end up with some great, cute ways to produce NADPH without the explicit need of transhydrogenase. So this is actually beneficial to do. Now ATP is expended in reactions, but nowhere near as much as NADPH. So transhydrogenase, this is used extensively in prokaryotes to produce NADPH at the expense of ATP yield. Now remember, ATP yields from glycolysis are theoretical, and that energy gets spent simultaneously by growing cells. So it's fair to ask, in a vacuum, how much energy in the form of ATP could you get from glucose, given the mechanisms and the thermodynamics of what an organism can do. But in reality, the proton motive force that's generated by electron transport, this gets spent in a lot of different ways. And so ATP yield is cells never reach their theoretical maximum yield of ATP because there are so many other processes that require proton motive force. And essentially you're spending ATP indirectly through things like transport, through things like rotating the flagellum, for example, or using transhydrogenase that I just showed you. So ultimately, if we're thinking about biosynthesis, we have to keep in mind that cellular catabolism and cellular anabolism are concurrent processes. And what that means is that these things are happening at the same time. And this is a fundamentally important thing to keep in mind as to why are different cofactors used? Why are different enzymes used to perform identical reverse chemistry? And this is why. So the cell needs to make decisions about generating energy and where to spend that energy. And so, when we think about bacterial growth, it's not that they're eating all of the substrate in a particular environment, cashing in all of that energy, basically putting all of that energy in the bank and then deciding to produce new cells. All of these things are concurrently happening. Okay? So, next time, we'll begin to start talking about a much bigger picture, and that is cellular growth. So, this is more or less the end of intense biochemistry, and I appreciate that you have gone through this with me, and I hope I've been able to help you make some connections between what's going on in terms of prokaryotic diet and how they can use elements of their biochemical pathways to build the components of a new cell. So, essentially being able to consume macromolecules to make other macromolecules. And so we're going to emphasize on prokaryotic growth when we're talking about cellular growth of microorganisms, and I'll take you through some population level growth models in the next lecture.